Assalamu alaikum, everyone. Can you hear me? All right, okay. So my name is Hiba Raheel, and I'm a fourth-year medical student. So today we'll be talking about DNA replication. We'll be going over the basics of DNA replication, and then we'll talk a little bit about the clinical aspect. Inshallah, it's like straightforward, not too difficult, and I'll try to make it as easy as possible. If you have any questions, please stop me right away and mute your mics or drop it in the chat below. I don't mind being interrupted, all right? Okay, so let's begin. These are the objectives. Okay, so let's start off by describing what a normal DNA structure looks like. So we know that a DNA molecule is basically double-stranded, right? So it has two strands that run opposite to each other, meaning that one strand is going to, let's say, run this way, just hypothetically, whereas the other strand is going to run this way. So a DNA molecule has a double helical structure, and the DNA strands, each strand is joined together by a special bond called the phosphodiesterase bond, okay? So the DNA molecule itself, what is it made up of? It's made up of smaller subunits. So we know that a protein is made up of amino acids. We know fats are made up of fatty acid and glycerol. So everything in the body, every polymer, every larger molecule is made up of smaller molecules. So similarly, DNA is made up of nucleotides. Think of nucleotides like amino acids, okay? But we will talk about their individual structures in a little bit. And these nucleotides are joined together by the 3,5-phosphodiesterase bond. What does that mean? That the 3 end, so the carbon number 3 of the nucleotide is bonded to the carbon number 5 of another nucleotide with this bond called a phosphodiester bond, okay? All right, so when the cells divide in eukaryotes, which means organisms with multiple cells, so not unicellular organisms, when the DNA divides, each daughter cell receives a copy of the genetic information. So every cell in the body has DNA inside of it, with a few exceptions, such as the RBCs, but almost every cell in the body has a copy of this DNA. The DNA, which is, as you can see here, it basically contains your genetic information. What your hair color looks like, what your eye color is supposed to be, how tall you're supposed to be, your skin color, you know, different things. So each cell in the body contains the exact same copy of the DNA. And how is that achieved? It's achieved through a process called DNA replication. So DNA replication is a process in which each chromosome is duplicated before the cell divides. So before the cell, the single cell becomes two different cells, its DNA is copied. The DNA, the number of DNA is copied, so it's replicated. And once it's replicated, then the nucleus is going to split and it's going to form two different daughter cells, each containing the same copies of the DNA. Okay, so that was for the eukaryotes, right? Now for the prokaryotes, so eukaryotes are those organisms that are unicellular, right? Prokaryotes we are prokaryotes. So we are multicellular organisms. In prokaryotes, the DNA replication occurs in a semi-conservative fashion. What that means is that once the DNA is split, so, you know, as I mentioned, the DNA has two copies, so it's a double helical structure. In order to replicate this DNA, we need to uncoil it. Once it's uncoiled, one strand stays the same and another strand is built on top of it. So the red strand is coming from the old DNA and the gray strand is the new DNA that's being built. So if you think about it, every DNA, every double helical structure has two copies. It has a newer strand and it has the older strand. So this is what we mean by semi-conservative replication. So each individual parental strand remains intact in one of the two new duplexes. All right, so before we move on, do you have any questions for me? Is everything clear so far? Okay, all right, so let's move on. All right, so now we're going to talk about the actual process of replication, okay? Before the cell starts to replicate, there's an area, a bubble, kind of a place that forms in between the double helical structure. This place, so here's how it looks like. So this is, let's say, the DNA molecule, okay? And what is going to start happening is that a particular area of this DNA is going to start opening up, okay? And it's going to become loose, and this area, the two strands of the DNA molecules are going to start opening. When they start opening, this place is called the origin of replication. It's in the name. So origin of replication is the area from which the replication process starts. So any specific area on the DNA strand is going to start uncoiling, and when it uncoils, it forms this bubble, okay? This separate site for the initiation of replication to begin. This area is called the origin of replication. So in a eukaryotic cell, so, you know, like bacterias, each chromosome may have hundreds or even thousands of replication forks, or origin of replication. So another name for origin of replication is also a replication fork. So in the exam, if you have this term, do not freak out, okay? So in eukaryotic cells, each cell can have multiple origin replication fork occurring. And so the division of DNA occurs in both sides. It goes either way. So if there is a picture here, yeah. So this is what it looks like. As you can see here, this is the origin of replication, or also called the replication fork. And as you can see, there are multiple origin of replication along the DNA strand, which means that there are multiple sites along this DNA strand that begin to uncoil. Once they begin to uncoil, that is where this is the start of your replication. So certain enzymes are going to come and attach here and here, and then they'll start reading the DNA molecule in both the directions, like this, okay? But the most important takeaway message from all of this is that what is an origin of replication or replication fork? It's basically the beginning point of the process of replication, where the two DNA strands begin to uncoil. And from here onwards, the replication occurs in both directions, okay? It's bilateral. All right, this is just a diagram. We went over this. This is an animation that you can watch on your own. I have a PDF version, so I don't think that this animation is going to play here. But basically, what it shows is that how the DNA begins to uncoil, the strand begins to uncoil, and the replication fork forms. And then from there onwards, DNA replication and uncoiling of the DNA occurs in both the directions, this direction and this direction. Okay, so once the replication, the origin of replication forms, we have something that is the replication fork that forms. So basically, replication fork and origin of replication is used interchangeably. But once these two strands, they open up and they begin to uncoil, you have a certain enzyme that comes and it starts to unwind the rest of the double helical strand, okay? So DNA strand, this is one DNA strand, as you can see here in orange, and this is the other DNA strand in red, okay? So a replication fork, or the origin of replication forms, and then this enzyme is called DNA helicase. Now, how can you remember this? So, you know, the enzyme called lipase, right? What does it do? It breaks down lipids, right? We have something called the protease. And what does that do? That breaks down proteins. So similarly, we have an enzyme called helicase. And this enzyme, it breaks down the double helical structure, or the two strands of the DNA. So kind of think of it as a zipper, okay? So this enzyme here is the zip, which is opening the two strands, all right? So then we have these special proteins that come and attach to the nucleotides that are exposed. And what they do is that they prevent the DNA from coiling again. Okay, so these are called single-stranded DNA binding protein. And again, it's in the name. The function of the protein is in the name. It binds to the single strands of DNA. All right, so the double helical structure, or the DNA helicase, is going to start moving in this way, and it will begin to uncoil the DNA as it moves along. Okay. Now the unwinding process of the double helical structure, or the DNA molecule, occurs, right? Now what happens is that as the DNA molecule is uncoiling, the part of the DNA that is ahead, so in front of this part that is being uncoiled, can sometimes become coiled, okay? So it kind of becomes knotted, all right? So this happens when the DNA helicase, which is that enzyme, is unwinding the DNA. So the DNA is becoming coiled, all right? Now, we need to uncoil it, because if this supercoil forms, then the DNA can be damaged, and the DNA helicase will not be able to do its function. So for that, we have two special enzymes, okay? We have type 1 DNA topoisomerase, and we have another enzyme called type 2 DNA topoisomerase, but this one is present in unicellular organisms. So these are present in bacteria, okay? So one more time, let's go back here and let's summarize it very quickly. So far, what we've discussed. So the DNA molecule is made up of two strands, and the structure is called, just a second, the structure is called a double helix, okay? Now, we need to replicate this DNA. We need to make more copies of this DNA. How will we do that when it's in this form? So a part of the DNA begins to uncoil, and this region is called origin of replication, or the replication fork. So it begins to uncoil or unwind. Think of it like two wires that are untangling, okay? So they begin to untangle, and then the DNA molecule is now opened in bi-directions, in two directions, okay? How is that done? By this special enzyme called the DNA helicase. It breaks the double helical structure of the DNA. And once the DNA molecules are being separated, so the DNA strands are being separated into two, we have these proteins called a single-stranded DNA binding protein. And let's say in the exam, you forget the name. I want you to think of their function. I want you to think of what they do. Every time you forget a name of a particular anything, so enzyme, muscle, nerve, vein, your best shot at remembering it is remembering the function. So in this case, what does this protein do? It binds to the single strands of the DNA and it keeps it open. So single-stranded DNA binding protein. As the DNA helicase is moving along the DNA molecule, the part in front of it can get coiled. So this part is called positive supercoiling. So it becomes coiled. And obviously we can't replicate this part of the DNA because it's coiled. So what do? We have two enzymes. Okay, so it's basically the same enzyme, but it has different variants. All right, there is one variant that is present in eukaryotes, and then there's this other variant that's present in prokaryotes. So the one that's present in eukaryotes is called type 1 topoisomerase, and it basically nicks it. It basically cuts the tangled bit of the DNA. And so then this DNA strand can be, then this DNA strand can start replicating from here. And then in prokaryotes, we have type 2 DNA topoisomerase, also called the DNA gyrase. Okay. All right. So here are some important drugs that you need to remember. So some drugs in the body act, when do we prescribe drugs? So we can give drugs for an infection, or we give medications to patients who have cancer for chemotherapy. So one of the agents or one of the drugs that we use to treat infections are called fluoroquinolones. Okay, so these are a type of antibiotics. Now, these type of antibiotics, they function on a specific area in the cell. So all the drugs that we will be talking about, they have a specific role to play either on the host cell, so the human cell, or on the bacterial cell. So the aim of prescribing antibiotics is to stop the replication of this bacteria, to stop it from causing damage and making us sick, right? So how do we do that? How do these antibiotics work? These antibiotics, they target a specific area, a specific enzyme in a bacterial cell. So this first antibiotic that we're going to talk about is the fluoroquinolone. Okay, so you don't need to remember the name right now. I mean, you're going to remember it anyway in second year or third year, but one of the name is ciprofloxacin and nalidixic acid. Okay, so there are types of fluoroquinolones. So fluoroquinolones are like a big family of antibiotics, right? So we have fluoroquinolones, we have penicillins. So they have different types of names. So nalidixic acid and ciprofloxacin are types of fluoroquinolones, and they inhibit the prokaryotic topoisomerase 2, which means that they affect this part of the DNA replication. So what happens is that when the DNA in the bacteria is supercoiled, usually topoisomerase 2 or DNA gyrase, it's another term, so don't forget that. They may replace topoisomerase in the exam, and they may put DNA gyrase, okay? So basically, this antibiotic, it stops the function of DNA gyrase, which means that this cell, this DNA molecule, is going to remain supercoiled. It's going to remain coiled. And so that is how it affects DNA replication. Once DNA replication is affected, the bacterial cell cannot replicate, and therefore it can die. It will stop replicating. It will get enough time for our immune system to act and kill the bacteria. Okay, so this is a very important function of a medication that you need to know. Okay, so that was for antibiotics. Now let's talk about cancer agents. So anti-cancer agents or chemotherapeutic drugs are certain classes of drugs that are given to cancer patients. So in cancer, the whole explanation behind the cancer is the abnormal replication of damaged and abnormal cells, right? So certain cells in the body, they replicate at an abnormal rate, and there are defected cells, okay? So the target of anti-cancer drugs or chemotherapeutic drugs is to stop the growth of these abnormal cells, okay? So an example is drugs like etoposide, teniposide, daunorubicin, and doxorubicin. Okay, so these are 4 kinds of anti-cancer drugs. Etoposide and teniposide, these are anti-cancer drugs or chemotherapeutic agents that act on eukaryote topoisomerase 2. Okay, so they act on this topoisomerase. Let's go back here, this one. Okay. All right. And daunorubicin and doxorubicin are again anti-tumor drugs or chemotherapeutic agents that are used to treat leukemia, so cancer of the blood cells. And what they do is that they go and they attach between the DNA molecules. So let's go back here and I can show you where they actually attach. So let's say these are the DNA molecules. So daunorubicin and doxorubicin would actually start attaching like this between the DNA nucleotides. So these are called the intercalating agents. They intercalate or they intervene between the 2 DNA strands and between the 2 nucleotides like this, okay? And they prevent the DNA from uncoiling. They basically glue these DNA molecules together so that the DNA does not replicate and the cell, the abnormal cancer cell, does not replicate and it's inhibited where it is, okay? All right, so now that we have talked about those, I would really suggest you memorize those names and the function because they do come in some questions. And not just that, but it's very useful knowledge for later on in year 2, okay? Okay, so let's move on to some other enzymes. Oh, before I proceed, is everything clear so far? Do you guys want me to repeat anything or go over something again? Do you have any questions? Okay. So I just see one response, so I'm gonna assume that's good. All right, okay. So let's proceed. Now, DNA polymerase. Okay, so we have so far talked about opening the replication, opening the DNA fork and opening the double helical structure. Now let's move on to actually copying the DNA. We have the DNA, it was double-stranded at first, now there are 2 single strands. Now let's focus on how to copy this DNA and make more and new DNA. So there is this enzyme called the DNA polymerase, okay? This enzyme is responsible for copying the DNA templates, all right? However, these enzymes can only read the parental DNA in the 3 to 5 direction, okay? So they only read in the 3 to 5 direction. And this enzyme, it synthesizes or it builds new DNA strands in the 5 to 3 direction. All right, so what does this mean? So for those of you who don't know, the DNA molecule has a 3' end and 5' end, okay? Think of it as right and left, but for DNA molecule, okay? We can't really say right and left in DNA, right? So we use the terms 3' and 5', okay? Every DNA molecule has opposite. So as you can see, the 5' end is attached to the 3' end, right? And the 3' end is attached to the 5' end. So a DNA polymerase, which is now going to come and read this DNA strand, is going to read it from the 5' to 3 direction. My bad, from the 3 to 5 direction. So it will start reading it from here and it will go towards the 5 end, okay? So it will attach here and it will read this way. Once it's reached here, that is when it's going to start adding new nucleotides and start making this double helical structure. So it makes the new nucleotides and the new DNA molecule from the 5 to 3 end, but it reads the DNA from the 3 to 5 end, okay? So again, very important. So a DNA polymerase reads the nucleotide sequences or reads the DNA molecule from the 3 to 5 end and synthesizes the new strands from the 5 to 3 end. So the DNA polymerase, it basically catalyzes the elongation of the new DNA, all right? It builds the new DNA. So 2 jobs of DNA polymerase, not only reading, but also synthesizing, okay? It reads from the 3 to 5 direction and builds from the 5 to 3 directions, the opposite direction. That's why the DNA molecules are opposite to each other. Like they are complementary. They are exact opposite of each other. Okay, now let's talk about the RNA primer and the RNA primase. So the DNA polymerase, it cannot begin the synthesis of the new strand, okay? It only builds, it only reads. We need something to come and attach the very first molecule of DNA. So the very first nucleotide that is attached is done by the RNA primer. Think of the RNA primer as like a glue, okay? Or like the primer in your makeup routine. It basically is kind of something that you put at the base. That's the first thing you put on, okay? So the RNA primer is the first short double-stranded region that contains of an RNA base paired to the DNA template. So this is the very first nucleotide that is going to come and attach to the DNA molecule. And then on top of that, you will have more and more nucleotides, okay? So the primer is basically very short. It just contains about 5 to 10 nucleotides, not very long, and it comes and attaches on the 3' end of the DNA strand, okay? And what makes up this DNA primer? The primase, okay? All right, so now let's bring up all our knowledge, and now let's combine it and read this diagram. So these are the 2 double helical DNA strands. Okay, this is the 3 end, and as I told you, that DNA molecules or DNA strands are in opposite to each other. So 5 end is going to be joined to the 3 end, and the 3 end is going to be joined to the 5 end. Okay, we have something called the helicase, the DNA helicase, which opens up the DNA molecule. Once it opens it up, we have something called the topoisomerase, which cuts and fixes the supercoiling of the DNA molecule. Once that is done, we have something called the RNA primer, which comes and attaches to the DNA strand, and it begins the process of it basically makes room for the DNA polymerase to act. Next comes DNA polymerase. It's this big enzyme which starts reading the DNA strand in the 3 to 5 direction. This is very important, and it synthesizes the DNA molecule in the 5 to 3 direction. All right, so again, this is a short animation that you can watch in your own time. Okay, all right, so is everything clear so far before we move on to the other part? Okay, great. All right, so now now let's talk about some very important terminologies. Okay, so we mentioned that a DNA molecule in eukaryotes, so in multicellular organisms, is made in a semi-conservative pattern, right? So you have one molecule that is from the old DNA, and you have one molecule that is new, and you form it, right? So the old DNA is the one that is basically being separated. So let's try seeing here. Do we have an image? Yeah, so this is going to be the older DNA, okay? And then the new DNA, we will start forming it here, okay? So the new strand that is built, another name for it is called the lagging strand, okay? So new strand is called the lagging strand, and the DNA polymerase must work in the direction away from the replication fork, right? Because obviously the replication fork is the place where it's going to start, the DNA polymerase is going to start acting. So it needs to go away from it. And remember, the lagging strand is the new strand. And this is a very favorite question of Dr. Hannah's. So you might want to remember this. Now the lagging strand is synthesized in little bits, okay? So it's synthesized bit by bit in a series of segments. So you have a segment of DNA that forms, then another segment, then a fourth segment, and these segments are called the Okazaki fragment, okay? The Okazaki fragments are basically just nucleotides, a bunch of nucleotides that come attached to the parental strand, and these Okazaki fragments form the new strand, okay? And now we need something to glue these strands again. We need something to hold the Okazaki fragments together. And what is that? The glue DNA ligase. Okay, so DNA ligase, it just joins the Okazaki fragments. So let's pretend like this is the DNA molecule, and this is the new DNA molecule that is forming. So this is, I'm drawing the lagging strand here. And so the function of the DNA ligase is basically to attach the fragments like this and to form this entire strand and this entire new strand, or the lagging strand. All right, so this is what it looks like. This entire thing here is the bubble that I mentioned, the origin of replication, also known as the replication fork. It starts to uncoil in both the directions. So your lagging strand is the new one that forms, and the leading strand is the older one, the old strand that forms, okay? All right, again, some animations that you guys can read in your own time. Okay, so now the DNA polymerase, so once the DNA polymerase does its job, just like anything, it may have mistakes, right? It may have copied some nucleotide incorrectly, or it may have made a mistake, okay? So thankfully, there is a very efficient process in our cells called the proofreading process, which is actually done by DNA polymerase itself. So the proofreading process is basically where the DNA polymerase goes and it corrects its mistakes, and it fixes all the places where there are incorrect nucleotides that have been placed. And it basically goes and it has this exonuclease activity, also known as kind of like a scissor kind of activity, to remove the mismatch or the incorrect nucleotides, right? So far we've talked about 3 functions of DNA polymerase. Not only does it read, it also builds, right? It also builds the DNA strand. And it has an exonuclease activity, which means that it has the ability to proofread the DNA molecule and to identify any mistakes and correct them so that they're not passed along to the new cells, because this is how mutations arise, right? As surprising as it is, the chances of developing cancer and developing mistakes in the DNA itself is very, 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 very rare. And that's thanks to the DNA polymerase. So because of its exonuclease activity, it has a 3 to 5 exonuclease activity, and it basically cuts away the faulty faulty nucleotides. All right. Now there's something at the end of the DNA, or at the end of the chromosome. So this thing is called, it's kind of like a cap, okay? This cap is called the telomeres. So the telomeres are basically non-coding and repetitive sequences of nucleotides, and they are present at the ends of the eukaryotic chromosomes. And what these things do is that with each round of replication in most of the normal cells, the telomeres are shortened, because remember I said that, you know, extra DNA, incorrect nucleotides are removed, right? So the telomeres with every round of replication get shorter and shorter and shorter and shorter. Okay, so the telomeres are shortened because the DNA polymerase cannot complete its synthesis. So unless the telomeres is not shortened, the DNA polymerase is not going to be completing its function, right? So this happens at the 5' end. So this contributes to the aging of the cell, because eventually what happens is that with every round of replication, every time in a person's body the cell is replicating, the telomeres are getting shorter and shorter and shorter and shorter and shorter. So the function of the telomeres is to protect the DNA. So every time they get shorter with replication, you can actually see that as the human ages, as the person ages, their telomeres become shortened. So some scientists claim or hypothesize that we can preserve, or we can stop the process of growth and aging in humans or animals or whatever if we preserve the telomeres, but you know that can't happen, unfortunately. So yeah, this is the whole thing that we just discussed, summarized. All right, so it's the same thing that I just told you about, that the telomere shortening have a correlation to human aging and developmental delay, right? So the decreased telomere length in some cells have shown a higher mortality rates in older patients, because with age, these telomeres grow shorter and shorter and shorter in size, which means that now the DNA is eligible, or the DNA is capable of undergoing damage, because this telomere was actually the thing that was protecting the DNA. So now this DNA can undergo damage, and it can actually, it has shown to have a higher mortality rates in older adults. Okay, and so there's a very interesting correlation between the telomere length and some psychological stress. And so they say that when they say that stress makes you look older, that is literally what they mean, that the stress basically, it makes these telomeres shorter, and that can like lead to psychiatric diseases and all of that. Okay, so thankfully in our body, we have an enzyme called the telomerase. Okay, so the telomerase is there to kind of, it's present in the eukaryotes, first of all, and it's there to kind of maintain the telomeres. Okay, so it just takes care of the telomeres. It replaces any sequences that would have been lost during the replication. But obviously, you know, with age, the number or the population of telomerase, the enzyme, kind of decreases. So the function of the telomerase is basically to maintain the telomeres and to make sure that not all of the telomeres is lost in every cycle of replication. So normal telomerase activity can only be found in like embryonic cells or productive cells or stem cells, right? Because these are the cells that basically give rise to potentially all the cells in the body, right? That is the function of the stem cells. The stem cells can form potentially into any other cell. The embryonic cell has the potential to form into several other types of cells. So we need these cells to stay alive for as long as possible. And for that, the enzyme called telomerase exists in them. However, in other cells, such as cardiac cells or brain cells or hepatocytes, we actually do not have telomerase. So all the cells that are not reproductive cells, that are not germ cells or stem cells, they're called your somatic cells, right? And they don't have this telomerase. Cancer cells, however, sometimes have a very high level of telomerase, which is actually very smart. If you look at a cancer cell, it's actually a very, very, very smart cell because it is very high in telomerase, which means that if telomerase is high in population, the telomers cannot become short. And the size of the telomers in every cycle of replication stays the same, which means that the cell is immortal. And that is exactly what happens in cancer cells. Every malignant cell, every cancer cell, the reason why it grows and it thrives and it, is not killed by all the defenses in our body is because it has this telomerase enzyme, which maintains the telomers. So lack of telomerase activity in the somatic cell results in the progressive shortening of telomeres with each cellular replication event. All right, so before we move on, do you guys have any questions? Do you guys want me to repeat anything? Go over something? Is everything clear so far? All right, great. Okay, so let's move on and let's talk about some nucleoside analogs. Okay, so basically, these nucleoside analogs are kind of like, they're like mimickers, okay? They are copycats of basically the original nucleotides. And the reason why they're there is because the DNA growth can be blocked by incorporating these analogs. So think of these analogs as like a full stop, okay? Or like kind of like something that stops the growth of the DNA molecule. So each DNA chain, once enough of it has been replicated, we have nucleotides that come and attach to it, and that's enough. The DNA has been replicated. Another way to stop this DNA from replicating further is something called the nucleoside analogs, and they're basically modified sugar portions of the nucleotides. So they block the DNA replication and they slow down the division, right? And this is essentially very useful in rapidly growing cells and in viruses, because this is when in viruses and growing cells, we need this nucleoside analogs to stop the growth of the DNA molecule. So the cytosine arabinocyte is basically another form of the cytosine nucleotide. And what this does is that it's acting as a chemotherapeutic drug or an anti-cancer drug, okay? And it does the same thing. It goes and attaches to the nucleotides, it goes and attaches to the DNA molecule, and it stops the growth of the DNA chain. Another very important molecule is the Adenosine Arabinosite, and this here is the antiviral agent. So it's very, very important in certain viral infections such as herpes, zoster virus. It's also used in hepatitis. And again, the function is the same: cytosine arabinocyte and adenine arabinosite. These are two nucleoside analogs, so they mimic the nucleosides, the actual nucleosides in the body, and they block the DNA replication by attaching to the DNA molecule. Okay, so here are some names of more nucleosides. So HIV, as you probably must know, is a viral infection, and it's a sexually transmitted disease. And so far there has been no cure for HIV, and HIV can only be, we can only stop it from progressing, but we cannot remove it altogether. right? So one of the two agents that are very commonly used in the treatment of HIV are azithromycin and zidovudine. So azithromycin and zidovudine. And these two are, again, antiviral agents, and they are nucleoside analogs, which means that they're copycat versions of the actual nucleosides, and they go and they attach to the hydroxyl group of the nucleotides, right? And they prevent the hydroxyl group from the third carbon of the nucleotide from being removed, okay? And that's how they stop the DNA replication of the HIV virus. All right, so now I have some questions for you. But before we proceed on to the questions, do you guys have any questions for me? You guys have no questions? Is everything clear? Is everything good? All right, okay. Someone's asking if I can please go over the HIV treatment once more. Yeah, sure, of course. Is there anything else that you guys want me to go over one more time before we proceed to the questions and then the next lecture? All right, okay, so let's just quickly go over the HIV treatment one more time. So the HIV is basically caused by a virus. And this virus, it basically decreases the white blood cell population in our body, okay? So this virus cannot be killed. Instead, it can be stopped. Once it goes in the body, once it has its effect in the body, you cannot eliminate it completely, but you can, however, stop its growth. So for that, there are very two popular drugs called azithromycin and zidovudine. These two drugs are basically copycats or nucleoside analogs of the actual nucleosides in the body, but they're not. Okay, they're not the nucleosides that are produced in the virus itself. They just look like it. They're just the copycat version. And what they do, their function is to essentially stop the DNA replication. So they go to the hydroxyl end, which means the OH end of the third carbon, so this one. They go to the OH end of the third carbon of the nucleotide and they bind there. And they basically stop the DNA replication from there on forth. So the viral cell actually stops, cannot replicate anymore. The DNA cannot replicate, and it just, dies. Okay, so let's go on to some questions. All right, so let me read this for you guys. So there's an investigator who is studying the biology of human sperm cells. Okay, and she isolates spermatogonia obtained on a testicular biopsy from a group of healthy male volunteers. She finds that the DNA of spermatogonia obtained from these men show a large number of TTAGG sequence repeats. Okay, so they're repeated. This finding can best be explained by an increased activity of an enzyme with which of the following functions. Now, remember, the question says that there are repeated TTAGG sequences on these sperm cells. Okay, so which one of the following would you think is defective? Do you guys have any answers? Okay. All right. Is that it? 2 responses only? So remember I said that, okay, F, right? Anything else? So focus on what type of cell she's studying. She's studying the sperm cell. Okay, so we talked about this very specific cell that is present in sperm cells only. And we said that this one right here is very important for the preservation of the age of the cell. Okay, so let's discuss this. All right, so the answer here is actually, okay, so before we go on to the answer, let's just break down the question itself, right? So the scientist, whoever, she's studying a sperm cell, okay, which is a germ cell, right? And it has this repeated number of DNA sequences, so TTAGG. So if you remember, we talked about something called a telomere, and we said a telomere is kind of like a cap that's present on the DNA, and it kind of is repeated sequence of the DNA, and it's named, and there are some enzymes called the telomerase, which are present in a few cells in the body, and they kind of preserve this function of the telomere, right? So this enzyme is basically found in the eukaryotic cells, right? And they have their own RNA template, and then they make their own, they basically maintain their own telomeres. Okay, and they synthesize the telomeres from the 3' end of the chromosome. And basically what these cells, these enzymes do is that they prevent the loss of the function of the telomere. They prevent the loss of this genetic material that occurs in every DNA replication. So the answer here is actually E, because it's the RNA-dependent synthesis of DNA. So this enzyme is basically protecting the synthesis of DNA. And there are high telomerase activity in normal germ cells, so in sperm cells, stem cells, embryonic cells. And we also said that telomerase activities can also be found in cancer cell. So what is the function of this enzyme? That's what they're asking, that increased activity of this enzyme. What enzyme are they talking about? They're talking about the telomerase. And what's its function? It's to basically do the DNA dependent synthesis of RNA dependent synthesis of DNA. Okay, so the second question, an investigator studying DNA replication of Campylobacter jejuni inoculates a strain of this organism into a growth medium that contains radiolabeled thymidine. After 2 hours, the rate of incorporation of radiolabeled thymine is measured as a proxy for the rate of DNA replication. The cells are then collected and centrifugation and by centrifugation and suspended in a new growth medium that contains no free uracil. Okay. After 2 hours, the rate of incorporation of radiolabeled thymine is measured again. The new growth medium directly affects the function of which of the following enzymes? All right, so let's break down this question and then you guys can answer, okay? So we basically have, they're basically just forget everything, look at the key important words. They're telling you that they're studying the thymine, they're studying DNA replication, and they have inserted radiolabeled thymine. So what they have done is that they've attached a special radio molecule to thymine, which is one of the nucleotide. And every time thymine is going to be incorporated in the DNA, we will know that, okay, this thymine has been incorporated, all right? Now, however, however, it does not contain uracil. So as you may have known that thymine attaches to uracil, right? So now uracil is not present. So 2 hours later, we're going to check how much thymine has been attached to the DNA molecule, right? So which one of the following enzyme is this entire process going to affect? So write your answers. So remember, we're talking about uracil, okay? We're talking about an RNA, not a DNA. So whoever said C, you would be correct, but we're talking about an RNA. So I'm gonna wait for like more people to answer. All right, so the answer is actually F. So good job for whoever answered F. You're correct here. And the reason why it's F is because remember, uracil is present in RNA and not DNA. So I know this thing was not part of my lecture per se, but I thought this was a really interesting question. And these are the kind of questions Dr. Aljada loves to bring, sort of like complicated and, very USMLE oriented. So uracil is basically an RNA complex. And remember, we said that every time DNA strand needs to be built, we need to have something called the RNA primase or the RNA primer. The RNA primer, like I said, kind of acts like this base, like this ground zero for the DNA polymerase to build on. And for that reason, the answer is primase. Okay, this is the last question for this lecture. So an investigator studying the replication of bacterial DNA with modified nucleotides, after unwinding the double-stranded DNA strand, forms a Y-shaped replication fork that separates into two strands. At each of these strands, daughter strands are synthesized. One strand is continuously extended from the template strands in a 5-to-3 direction. Which of the following exclusively associated with the strand being synthesized away from the replication fork? So this is very, very, very important to notice here what they're actually asking. So they're saying that we have a DNA, bacterial DNA, okay? Unwinding takes place, replication fork forms. The daughter strands are synthesized. One strand is extended from the 5-way direction, and they're asking which of the following strands is synthesized away from the replication fork. Okay, so elongation in the 3 to 5 direction, RNA primase, 5 to 3 exonuclease activity, synthesis of a short RNA, reverse transcriptase activity, or repeated activity of ligase. Okay, so I'll wait for a few more people to answer, and then I will explain it. How many questions are left? So we just have one more after this, and then we'll move on to the next lecture. It's the lagging strand, yeah. All right, so for those of you who answered F, very good job. I'm very proud of you. It is actually F. And the reason why is because the answer is repeated activity of ligase, okay? So the DNA synthesis of the lagging strand, remember, is away from the replication fork, okay? In the 5-to-3 direction. So the RNA primase comes and attaches to the lagging strand first, and the RNA, and so it's synthesized, the RNA primer starts synthesizing in the 5-to-3 direction, okay? In prokaryotes, we have something called the DNA polymerase 3, which then attaches to the primer and it starts manufacturing the segment of the DNA into 5-to-3 direction. So this starts happening and you have smaller DNA fragments called the Okazaki fragments with the RNA primers in between, and they start attaching, right? Then you have this enzyme called the DNA polymerase 1, which then replaces the DNA primers because we don't need the RNA primers, my bad. We don't need the RNA primers, so they start replacing it and they start removing it, right? And then the very, very, very last step is that the ligase comes and it attaches and links together all the Okazaki fragments, kind of like a glue, to form a complete double helical structure, okay? All right, so one last question. So an investigator is developing a new drug that inhibits DNA replication in Salmonella typhi, which is basically a type of bacteria, by selectively targeting DNA polymerase 3. Okay. So here they've shown you the basically the bacterial DNA right here, and they're asking you which one of the following labeled proteins is most likely to be inhibited by the new drug. Now remember, one thing that I didn't mention earlier was that DNA polymerase 1 is the one that's found in eukaryotes, and DNA polymerase 3 is found in prokaryotes, but it has the same function. It reads, synthesizes, excises. Okay, so which one of the following here is this enzyme which is activated, which is basically inhibiting DNA polymerase 3 is going to act? Where is it going to act? So the options are A, B, C, D, E, F, G. Okay, so for those of you who said A, excellent job, very well done. It is in fact A. The reason why it's A and not F here is because remember, it's going in the 5 to 3 direction. This is very, very, very important that it's moving in the 5 to 3 direction, and that is indicated by this arrow right over here. This is not, F is not this, because if you focus on it closely, this is actually ligase. This is removing the RNA primers, and Okazaki fragments. And it's kind of like gluing everything together. So the answer is A, because that's the one that basically builds and synthesizes the DNA molecule. Okay, so with that, we're done with this lecture. We have one more lecture, which is just 10 slides, very short. Everything that we've taken so far, but just like the clinical aspect of it, and I've included just 2 questions at the end. So we can either take like a short 5-minute break, come back, finish those 10 slides, and then we're done for the day. Or we can continue with those 10 slides right now. And yeah, we can just get it done with earlier, 5 minutes, 10 minutes earlier. So what do you guys want? Sure, but I need more people. Like I need more people to tell me what they want. You want to keep going? You want to take a break? It's up to you guys. You guys are the one paying attention. Okay, I have just 2 people responding. So like, 5-minute break. Okay. All right. So let's just take a 5-minute break. Please be back here by 9:27, 26 maximum, so that we can start the next lecture and be done with it. Okay. For those of you who want to join. Okay. All right, so let's go ahead and start our second lecture. So hopefully, inshallah, we'd be done by like the next half an hour. All right. Okay, so let's begin. All right, so now that we've talked about DNA synthesis and replication and all of that, we're now going to talk about some mutations, right? So as I told you in the previous lecture, there are several ways in which a body can actually prevent the accumulation of abnormalities, right? The accumulation of several types of mutations and DNA errors and all of that. So now we're going to talk about such types of like mutations. What causes these mutations? What causes these injuries to the DNA molecule itself? So one of the things that is very important, and I'm pretty sure that most of you must have heard of this, is the UV light, or basically the sunlight. And that's the reason why, they tell us wear sunscreen to protect yourself from skin cancer. So UV light is one of those things that induces dimerization of the pyrimidines. So pyrimidines are types of nucleotides, and UV light causes damage to these pyrimidines, right? And this is one of the main mechanisms for skin cancer. And another way in which the nucleotides can also be damaged is through radiation, right? And that's the reason why they say pregnant women or younger children should not have radiation, should not have X-rays, and you should not do an X-ray for every single patient all the time. You need to have like a valid reason or an excuse. So one of the reasons why is because X-rays contains ionizing radiation that causes literally opening and breaking of the strands of the DNA. So all these things can happen, and these are just like the tip of the iceberg. There's so many other insults that can happen to the DNA molecule that can damage the DNA molecule. However, our cells have a very concrete repair mechanism for these such types of modifications, right? But sometimes such damages to the DNA go by unnoticed and they cause mutations. So the accumulation of mutations is basically what causes the arise of cancer and aging, right? These mutations, they accumulate, they pile up, they collect inside a cell, and then ultimately, once they accumulate intensively inside a DNA cell, the cell will now become abnormal. So it's not just the accumulation of one mutation, it takes a lot of mutations to the DNA to cause a typical cancer cell to form. Okay, so there are multiple repair systems that allow the stability of the genome, and any defect in the DNA repair just adds on to the likelihood of that cell developing into a cancer cell, okay? So one of the things that we're going to talk about is damage to the thymidine nucleotides. So this kind of damage is called nucleotide excision repair. So if you go back and go back to when we talked about the function of the DNA polymerase, we said that one of the functions of the DNA polymerase is to remove or excise any abnormalities, right? So what happens in this whole scenario is that the ultraviolet light or the sunlight basically induces the formation of dimers between two thymidines, right? So the thymidines, they basically like form, so they become really attached. They basically are glued together in the DNA, okay? And this formation of thymidine dimers interferes with DNA replication and normal gene expression, because these abnormal thymine nucleotides are kind of like super glued together. They cannot be eliminated by the nucleotide excision repair mechanism, which is done by the DNA polymerase. So what happens is that these DNA dimers, they remain in our genome. They remain part of the DNA. And as a result, they are copied. They form this kinking of the DNA molecules. As you can see here, the two adjacent nucleotides, so they were like TNT next to each other. So this is what it looks like in the normal cell. As you can see, TNT is next to each other. And then because of ultraviolet light or some abnormality, this TNT molecule, so tyrosine and tyrosine, they kind of like link together and they form a bond and then form dimer. Okay. And when that happens, that literally happens between two adjacent, not parallel, adjacent, like right next to each other. Okay. Parallel would be on the other molecule of the DNA, but we're talking about two tyrosine adjacent molecules that kind of like get stuck together and they form this kinking in the DNA molecule. And as a result, you have this pathology called the xeroderma pigmentosum. So xeroderma pigmentosum is basically a genetic disease. And by the way, guys, this is very, very, very rare, but somehow it has a significant relevance to biochemistry. And this has been a favorite topic of questions ever since like my batch, so ever since 11th batch. And so this disease arises from the abnormality of the dimerization of the tyrosine molecules, and this forms a mutation. And this is basically defective repair of the thymidine dimers. This is an autosomal recessive disorder. Again, very, very, very important because she loves bringing this. So autosomal recessive disorder. And sorry, so I'm going to put a red star here for you guys to know that this is extremely important. And it's characterized by hypersensitivity to sunlight, because every time this patient goes in the sunlight, their DNA starts getting damaged and these mutations accumulate. They have skin freckles, ulcerations, and eventually in severe forms, skin cancers, unfortunately. And these patients, they unfortunately don't have a very long lifespan. And because of the development of carcinomas and melanomas, which are a form of skin cancer, they appear early on in life and the patients die, unfortunately, because of the skin cancer. So one of the ways to like prolong life and to limit the amount of skin cancers in these patients is ask them to avoid exposure to sunlight or UV light. And yeah, so again, this is in very severe cases. So that's another reason why this can even happen to like us, right? So this is another reason why they tell us to wear sunscreen. So again, very important illness or disease to know about. Know the mode of inheritance. This is autosomal recessive, okay? What is the default in this? Like, what is the whole problem? It's the nucleotide excision repair. So nucleotide excision repair, the tyrosine dimers are not, the thymine dimers are not excised, okay? There's a problem in the excision, in the removal of these thymines, which basically leads to this unfortunate illness. Now, another thing that we're going to talk about, another defect, is a mismatch repair. What happens if a nucleotide is matched to the wrong DNA molecule, right? So what happens if guanine is attached to, I don't know, adenosine, right? So this is what we're going to talk about now. So sometimes the replication errors, they tend to escape from the check mechanism, proofreading mechanism, the checkpoints in the DNA synthesis, right? And this causes a mismatch of one or more bases. So any strand that contains the mismatch is in normal circumstances identified and it's removed by exonuclease. However, sometimes it's not removed, okay? And this mismatch proteins, they are kind of like, because of maybe some genetic problems, they're not removed and they accumulate. These mutations, they accumulate and they cause something called non-polyposis colorectal cancer, also known as Lynch syndrome. So basically in Lynch syndrome, these patients have polyps all over their colon. So inside their colon, it's filled with polyps, like thousands of polyps, thousands. You can try Googling this, try searching Lynch syndrome colonoscopy, and you will see thousands of polyps in the colon. And this is because these polyps form because there's abnormality in the wall of the mucosa and the cells grow at an abnormal rate, which causes these polyps to form. And a polyp in the colon can most of the time be actually translated to cancer, that can convert into cancer. So every time we see a polyp, most of the time we remove it, okay? But in this disease, in hereditary non-polyposis colorectal cancer, and the short name of this is Lynch syndrome, this patient has hundreds of polyps. However, normally, I mean, if you pick from 5 people, if you pick one person, one of them is going to have a polyp. I may have a polyp, you may have a polyp, and that's normal, that's fine. But these patients have thousands of polyps, okay? And so this can predispose them to colorectal cancer. And only about like 5% of colorectal cancer is formed through the mutation of mismatch repair. Okay, so these are just the 2 things that she has explained here. And that's it, like a very short lecture. All right, so we just have 2 questions and let's just do them and then we're off, okay? So a 5-year-old girl is brought to the physician by her mother because of a 1-month history of a painful ulcer on her face. She has developed painful sunburns in the past with minimal UV exposure. Examination of the skin shows a 2-centimeter ulcerated nodule on the left cheek. There are scaly hyperpigmented papules and plaques over the skin of the entire body. Ophthalmologic examination, you think I know how to pronounce that, shows decreased visual acuity, clouded corneas, and limbal injection. Examination of a biopsy specimen from the facial lesion shows poorly differentiated squamous cell carcinoma. So it's a type of cancer. Impairment of which of the following proteins is the most likely cause of this patient's condition? So as you can see, there's like a lot of like here and there and like a lot of stuff, but the most important things let's highlight are painful ulcer. Okay, so she has a painful ulcer. She has, it comes after sunburns and minimal exposure to UV. There's this ulcerated nodule, scaly hyperpigmented papules, plaques. She has also one more thing, squamous cell carcinoma. She has cancer. So all these things point to xeroderma pigmentosum. And as you guys mentioned, excellent, very good. The answer, in fact, is A. Okay, why? Because this is a problem with the excision of the thymine dimers, and so they are not excised, and that's what causes excision. That's what causes xeroderma pigmentosum. So very good job. Okay, another question. An investigator studying DNA repair process, and the investigator inactivates a gene encoding a protein that physiologically excises the nucleotides from damaged, bulky, helix distorting DNA strands. A patient with a similar defect in this gene is most likely to present with which of the following findings? Hyperpigmented macules and aplastic anemia, malignant breast and ovarian growths, dry skin and increased photosensitivity, colorectal and endometrial cancers, leukocoria, and painful bone mass, ataxia, gait, and facial telangiectasis. I know there's like a lot of, a lot of words that you don't know, but just focus on the things that I've highlighted. So the problem is basically she's studying a gene that codes for the excision repair. So an enzyme that does excision repair. And if that is abnormal, we talk about xeroderma pigmentosum, right? So one of the characteristics of xeroderma pigmentosum is xeroderma pigmentosum arose from basically thymine dimer excision repair. So excellent. All of you who've said C, that is in fact the correct answer, because those are the characteristics of xeroderma pigmentosum. For those of you who said D, colorectal, that is as a result of faulty mismatch repair. So in mismatch repair, basically there is abnormality in the linking, and the right nucleotide is not linked to the DNA molecule, which results in something called Lynch syndrome or hereditary polyposis, and that causes cancer, colorectal cancer. All right, so that's it, you guys. Thank you so much for attending, and you guys can email me if you have any questions. I'm going to write my email here in the chat. And so you guys can email me if you have any questions. Good luck for your final. Oops, sorry, I think I emailed it. Wait just a second. Good luck for your final. You guys are nearly there. And yeah, hope to see all of you in second year, inshallah. Bye.